Hello and welcome students. In this video, we are going to take a look at the complex process called glycolysis. And basically, it's made of two words, glyco and plus lysis. So glyco stands for sugar, and then lysis means to split apart. So basically, glycolysis is the process of splitting apart a glucose molecule. Also, it is a universal pathway, goes from yeast to mammals. It's a common metabolic pathway that happens. Hence, we are going to study this deeply. Now, basically, what happens is that, as you know, glucose is a six carbon molecule. So, when it undergoes glycolysis, it breaks down into two pyruvate groups. And the pyruvate is basically a different chemical formula with three carbons on each one of them. So, that is the main overall idea about glucose being broken down into two pyruvates, and hence the process of glycolysis. Now, the energy is released from glycolysis is in the form of ATP. Remember, ATP is adenosine triphosphate, the energy currency of the cell, and NADH. Now, the next point is that glycolysis takes place in what place does it take place? Cytosol of the cell. All right, so now for point number 7, let us take a look at the actual reaction that happens. So basically, we have the glucose molecule, we have the NAD plus, NAD ion, plus 2 ADP, 2 of adenosine diphosphate, plus the 2 phosphate ions. Now, they will, after glycolysis, what is the end product? The end product is the 2 pyruvate plus 2 NADH plus 2 hydrogen ions plus 2 of molecules of ATP, adenosine triphosphate, and plus water is also released. So let us take a look at this is the 6 carbon glucose, and then this is the diphosphate. Now here we have the 2 pyruvate. The pyruvate have 3 carbon, and then ATP is the energy currency of the cell, plus water is released as well, and the hydrogen ions. So this is something that you need to remember. This is important, the reaction. And then for your course, we will delve deeply into as to what are the steps, the total of 10 steps for glycolysis, and we'll go and take a look at them one by one. All right, so now let us take a look at what are the two main phases or how we break down glycolysis into 10 steps and what happens in phase 1 and then what happens in phase 2. So the very important thing to remember here is that the phase 1 is called the investment phase. Now, investment is basically like when you are putting some money into savings account or investing some money somewhere. So in the same way, the energy of the cell is being invested into this process. Hence, it consumes energy, the second point. Next is, when does this happen? It happens in step 1 to 5. And then 2 ATP molecules are used as a part of this phase 1. Now, because it's consuming energy, it is called an endothermic reaction. Now let us take a look at the phase 2. What happens in the phase 2? Phase 2 is basically the payoff phase. Now phase 1 has happened. In the phase 2, we are going to get energy. So it's producing energy, and it's happening from step 6 to 10 that we'll take a look at today. So payoff phase is the name, and then 6 to 10 steps. Now, what is released as a part of phase 2 reactions? 4 ATP and 2 NADH molecules. And it is also exothermic because energy is produced. Now, what is the net gain of glycolysis? Net gain will be basically, we have already used these 2 ATP molecules. So in the end, we are getting 4 ATP. So our net gain is going to be, after glycolysis, 2 ATP and then 2 NADH molecules. So that's the net gain of a glycolysis reaction. So now we will take a look at the step 1 for glycolysis. And step 1, we are going to take a look at the chemical formula of glucose. Now in the chemical formula of glucose, you can see here that we have a hydroxyl group. So after this first step is over, we end up getting the hydroxyl group is being replaced with a phosphate group. And that is what you need to remember for the first step. So let us label the carbons here, 1, 2, 3, 4, 5, and then this is the 6th carbon. So in order to write its name, we will still call it the glucose. It's still the glucose molecule, glucose 6-phosphate. will be the name because this hydroxyl group has been replaced with the phosphate group. So an important point to remember is that the hydroxyl group is replaced by a phosphate group. Now we are still continuing with step 1. As you saw, the hydroxyl group was replaced with the phosphate group. Let us take a look at it again. You know, the hydroxyl group is replaced with the phosphate group. And basically the addition of phosphate, as you saw in the last video, is phosphorylation. That's the name of the process. And where does this extra phosphate group come from? It comes from ATP. And then basically what we have is the ATP, and there's an enzyme that helps with this process. We end up getting an ADP and an extra phosphate. So diphosphate is this, and this one is the energy currency of the cell, triphosphate. Now there is a special enzyme that helps with this, and the name of the enzyme is hexokinase. So this enzyme basically works and helps in the working of the ATP to ADP, releasing the extra phosphate group, which is then used in step 1. So now let us take a look at the step 2. Now, if you remember from step 1, the product of step 1 was glucose 6-phosphate. So that's the same thing we have right now, the glucose 6-phosphate, because this is the 6th carbon. And what happens is basically we get the same chemical structure formula, but the structure is different. Here we have fructose. So how will we write the name of this one? Fructose 6-phosphate. This is the 6th carbon here. So the name will be. Now, as these both of them are isomers of each other, the enzyme that helps with this process is called isomerase. And this is a reversible reaction. Hence, you can see the arrow is pointed both sides. So just remember that in step 2, we are changing from glucose 6-phosphate to fructose 6-phosphate. Something important to remember. All right, so for step 3 for glycolysis, what do we have here? Now here you can see that the reactant of this reaction is basically what we had in the last step, fructose 6-phosphate. So that is what we are moving on to. So we'll call it fructose 6-phosphate. And then basically what is happening here? You can see that we had one phosphate group, but after this reaction, we are ending up with 2 phosphate groups. So the name will change accordingly, but it is still fructose. So the process is phosphorylation because another phosphate group is added. And let me erase these names because I just want to name it according to the carbons. And here we have the first one, second, third, fourth, fifth, and sixth. So how will the name change now? The name for this fructose will be fructose 1 and 6. Both these places we have the phosphate group. And how many phosphate groups do we have in total? 2. So biphosphate. So just remember here we just had one phosphate group. After phosphorylation, we got 2 phosphate groups in the products, and hence the name. And basically, where does this extra phosphate comes from? The extra phosphate comes from ATP. All right, so the products of the last step are going to be the reactants of this step now. So now let us take a look at what was the product for the last step. What was the name of it? The name was fructose 1,6 biphosphate. That is the name I'm going to add here, fructose, 1,6 biphosphate. Now, what is happening here is basically a totally different reaction, and we'll end up with two products here. This one is acetone, and the name of this reaction, this product is glyceraldehyde 3-phosphate. Now, why 3-phosphate? As you can see, we have first carbon, second carbon, third carbon. So that is how the name comes 3-phosphate. This is the formula for acetone. These are the two products we are getting. And remember, this was what we started with, fructose 1,6 biphosphate. The enzyme that helps with this reaction, this is a reversible reaction, is called aldolase, the name to remember. All right, so for step number 5 for glycolysis, on the left-hand side, we have dihydroxyacetone phosphate. This is the reactant here. Again, a reversible reaction. We can move between both of these. And then on the right-hand side, after the help of an enzyme, we get a product with the name of glyceraldehyde 3-phosphate. 3-phosphate on the third carbon, you have the phosphate group. Now, what is basically happening? There's something important to remember about these two reactants and products that we have. They both have 3 carbons, correct? They have 5 hydrogen atoms. They also have 6 oxygen, and they have 1 phosphorus. Now, if you take a look at this, their chemical formula is not changing, just the structure is changing. So when this happens, both of these are isomers of each other. So isomers. So remember, anytime we have isomers being formed, the enzyme that helps is isomerase. So hence, isomerase is the enzyme that is helping with this reaction. All right, so now we are moving on to the payoff phase. That means this is the time that all the energy currency is going to pay off as we are coming to the last half of the metabolic pathway glycolysis. So basically here, what do we have? On the reactant side. On the reactant side, you can see that this is the third carbon that has the phosphate group. So we will have glucose 3-phosphate, plus we also have an inorganic phosphate. Now remember, this is a reversible reaction, can happen both ways. And what do we end up with? Now here we'll number the carbon 1, 2, 3. So we have the phosphate group here and here as well. So what do we call it? The name will be 1,3 bi. So we have 2 phosphate groups. So biphospho glycerate. Now, as you can see, as the reaction is moving forward, this hydrogen is removed, and in place of the hydrogen, we get the phosphate group. So that is something you need to remember. And the enzyme that is helping with this reaction is called dehydrogenase. Let's write the name here also, dehydrogenase. And why is it called dehydrogenase? Because hydrogen is removed and a phosphate group is added. Hence the name dehydrogenase. All right, so now we are on step 7 of glycolysis. Long steps, a lot to remember in this concept. And basically, here on the reactant side, what do we have? I numbered the carbons for you, so it's easy to figure out the name. So this one is going to be called 1,3 biphospho-glycerate. And then this is a reversible reaction. And on the right-hand side, what do we have? Let us label the names here. the carbons here, 1, 2, 3. So here you can see pretty much everything else is the same. The only thing is we have lost one phosphate group. There were two phosphate groups here, and we just have one phosphate group on this side. So we will call it, on what place is it placed? 3-phosphoglycerate. So everything else remains the same. And then we just have the placings of phosphate are same as well, just the one phosphate that we lost. Now, where did we lose this phosphate to? Where did it go? Basically, this phosphate has been used with the ADP to give result to ATP, which is the energy currency of the cell, adenosine triphosphate. So where did the phosphate group go? And then the answer for this one will be, it is used with ADP diphosphate plus the phosphate group to give us ATP triphosphate. And then this process is also called substrate level phosphorylation. And the enzyme for this reaction is going to be called phosphoglycerate kinase. All right, so let us take a look at the step 8 for glycolysis. And step 8 is basically we had 3-phosphoglycerate in the last process, as you can see. We had the 3-phosphoglycerate. We are going to have that as our reactant, and it's a reversible reaction. Now let us label first carbon, second carbon, third carbon. Now basically here you can see pretty much everything is the same, so we can say that these are isomers. But this phosphate group has been moved from position 3 to position 2. So a mutation has occurred, you can say. So how will we name it? We will call this 2-phosphoglycerate. Everything else remains the same because it's at the second, with the second carbon now, that's why it's 2-phosphoglycerate. And the enzyme that helps, remember, which is the enzyme that helps us convert isomers into each other of their forms, is the special name is isomerase. So the same enzyme is used here again. And then what we need to remember here is that the phospho group has moved from third carbon to the second carbon. All right, we are coming now to step number 9 for glycolysis. And then as you can see on the step back, we have 2-phosphoglycerate. So that is what we are beginning with. 2-phosphoglycerate, that's the name for this one, because the carbon is second where we have the phosphorus group. And then it's a reversible reaction. And then the name for this one is phosphoenol pyruvate. Now, basically what is happening here? What are the changes that we are seeing from our reactants to products? So here you can see that we have this hydroxyl group and a hydrogen. Now this hydroxyl group and hydrogen will form water molecule, H2O. And then we are ending up with pretty much the same thing. Other than that, there will be water. So let us remember that we are losing an OH and a hydrogen to form H2O. And then we also call it a dehydration reaction. As you can see, there is a water molecule that is removed. And the enzyme is called enolase. And you will see why the enzyme is enolase, because we are working with an enol group. In chemistry, you must have studied. So this is the enol group that we are working with. So hence the name of the enzyme is enolase. Let's write down the name of the enzyme here, which is helping with the reaction. All right, so we are now on the step 10 of glycolysis, a pretty complex concept, and this video is long. So step 10 is basically the last step where we have phosphoenol pyruvate. Remember, we had phosphoenol pyruvate as our product on step 9. So we are using that, and then now we are also using magnesium and potassium ions. Now, what is happening here basically? What you need to understand is that this phosphate group is going to be lost as the part of this reaction. Now, where is it being lost to? Now, it is going to be actually added with ADP, diphosphate, to give ATP, which is again the energy currency of the cell, and hence energy is released and it is an exothermic reaction. So anytime we are working with the phosphate group, the enzyme is always going to be kinase. So that's the name of the enzyme. And we are losing the phosphate group is what you need to remember. And the name of the enzyme is kinase. And here the end result is also called a pyruvate, the 3-carbon group called pyruvate. So hopefully the 10 steps are pretty much clear to you. Basically, we are breaking down the glucose into different steps, and it is happening as a universal pathway in, pretty much all living organisms, and hence the importance of studying it. So thank you for watching the video. I'll see you in my next one. Have a good one. Bye.